Pollen. You've heard of it, you might be familiar with it in passing as that yellow plant dust. You might have learned about it in an early biology course. Maybe you haven't thought about it much since then, except as an annoyance in the spring and summer, especially if you have an allergy. For all its subtlety and the ease with which we can overlook it or even forget about it in the day-to-day, the existence and performance of pollen is nevertheless an integral part of how we live by its virtue of its agricultural necessity. Let's take a closer look. There's a common misconception that pollen is itself plant sperm, and while it is related and it is a necessary part of plant fertilization, pollen is the source and vehicle of sperm. It's not a gamete, but a gametophyte. In anthers, which is the male part of an angiosperm's reproductive structure, flower, the pollen mother cells undergo meiosis to give haploid daughter cells. These haploid cells then divide again, this time mitotically, each forming bicellular pollen grains. Of these two cells in a grain, one of them will be the tube cell and the other will be the generative cell. When a pollen grain germinates, the tube cell will push through this outer layer, the exene, and elongates, growing through a new tip. The generative cell will divide into two sperm cells. Pollen tubes can go very quickly and very long. They need to be long enough to go through the stigma, down the style, and into the ovules so they can deliver sperm to and fertilize an egg. If a grain of pollen isn't viable, or it doesn't properly germinate and develop, or there's something else wrong with it along a similar vein, fertilization can fail. While there are crops and some specific parthenocarpic cultivars that don't require fertilization or any pollen transfer in order to fruit, and there are many other crops that we just don't grow for their fruits or seeds in the first place, the majority of horticultural activity requires viable pollen and its successful development. Even if a particular crop doesn't require full fertilization, they might still require pollen transfer as part of their fruit and seed development process. Even if it doesn't require cross-pollination and it's self-fertile, a fruit still needs their own pollen to be working right. Sexual reproduction requires functional pollen. Therefore, conventional breeding and crop improvement methods also need it. Aside from a handful of exceptions, fertilization is a requirement for seeds to even develop, let alone be viable. Fruiting might never start. Fruitlets might abort early, seeds might never develop. Without proper pollen function, there's yield loss. Temperature can shape pollen performance. Regional and global climate change has brought more excessive heating during key periods for crop growth and development, which has hurt yields. This is a present and future problem. With this in mind, my project this summer through the Envision program, working in Dr. Beltran's lab at the Plant and Soil Science Department at UD, is built around in vitro pollen germination studies. There are two primary components that I've planned. First is developing a standard protocol for germinating tomato and Arabidopsis pollen, and then using that to, second, study the effects of increased temperature and treatments of abscisic acid, which is a plant hormone associated with plant thermal responses, on pollen sample germination. I'm still actively working through this first part. I'm far from the first person to seek in vitro pollen germination tools. Being able to induce and directly monitor pollen germination without the practical and literal obstacle of needing a pistil to pollinate and grow through reduces barriers to studying these microscopic objects. The two model organisms I'm working with, Arabidopsis thaliana and tomatoes, both already have numerous recipes for germination media from across decades. The goal of my work now is to identify and test a procedure for pollen germination that can then be used as a standard, routine practice in this lab in future projects. I don't need to invent a unique new media formulation from scratch, nor does it make much sense to try to start there. I just need to find something, make sure it can be efficiently and consistently be used in future projects, and record that. This has been an iterative process. Finding recipes, trying them out, making adjustments, trying to troubleshoot different things like sample collection, pH, storage issues, counting methods, data collection methods. It's a lot of troubleshooting. I haven't gotten a protocol that I'm really that happy with down yet, so I keep going.